Scientists have warned that reductions of up to 30% in the Earth's protective ozone layer could occur over Britain within the next...
In Brazil, thousands of square miles of rainforest are being cut down and burned. The amount of smoke from the fires is so great that it's even threatening to alter the world.
Every day, an area of tropical forest the size of the county of Kent is felled. Scientists now agree that the environmental costs of this destruction are literally intolerable.
The government's under growing pressure to protect the countryside from acid rain by cutting sulphur dioxide emissions from power stations.
Damage by acid rain is more widespread than previously thought. Some of Britain's most cherished landscapes have become damaged.
It's vital for us to understand our environment, to know what we're doing to it, and to learn how we can look after it. We can do this thanks to investigations carried out by scientists. Almost all scientific investigations, and not just those to do with the environment and ecology, have one thing in common: the basic methods used to investigate the subject, the methodology. So what are the secrets to success in science? Scientists seek ways to understand what they see. Sometimes they might have a theory or hypothesis to explain their observations, but even if they don't, they'll try to find out as much as possible before setting up tests, recording what happens, and drawing conclusions. And then so that others can learn from their work, they must communicate all of this to other scientists. We're used to seeing conferences of politicians, but meeting and talking like this is one of the key ways in which scientists communicate. It's often in settings like this where scientists will announce their latest discoveries. But for one group of teenagers, there's another reason why this conference is especially exciting. In just under 1 hour's time, they're going to announce the winners of the competition to find the young ecologist of 1994. Somewhere, there's a sealed envelope with the decision.
Projects arrive from all parts of the UK, which was just what we wanted. The other good part about the competition was that pupils responded from urban areas as well as rural areas, so that was great.
Oh, they varied from everything from conservation activities in woodlands to studying earthworms, decomposing litter, to seed banks in soils. We had a lot of initiative shown.
If we can get young children to start studying ecology from a scientific point of view, then the methods that they learn and the understanding they gain is going to be of immense value as they grow older.
The judges were looking for projects that use good scientific method. They chose 3 finalists. 5 girls from Devonport High School investigated plant and animal recolonisation. Stephen Dyke looked at the effect of acid rain on seedlings, and Ian Rushton carried out an investigation into earthworms. But more of these later. To find out just how important methodology is, I visited a research project where they have a great deal of experience in conducting scientific investigations. But first, I had to do some investigating of my own to find out what this building, called a rhizotron, is. Malcolm, what is a rhizotron?
Well, it's a rather grand name for a laboratory that allows us to look at roots. It comes from 2 Greek words, rhiza meaning root and tron meaning machine, so this is a root observation machine. But we use it not just for looking at roots, but also for looking at the animals that live in the soil and around the roots.
It's a very elaborate piece of equipment, but what makes you go to such lengths to study life underground?
Science is an activity that springs from curiosity, and what you should do is what interests you, what seems to you important, what seems to you to be answering questions and for increasing knowledge. And so at this stage, what we're really particularly concerned with is observing, is trying to see what happens in the real world. Once you know what is happening, then of course a lot of questions occur. Why is it happening? And as soon as you start asking why is something happening, then you put up ideas, hypotheses as to why you think it's happening. Then you can start testing them. But at the moment, we're still just trying to find out what this new world is like.
Can we go and have a look?
Sure. Show you. What's the steps? There you are. we're now looking at one of the windows, and through the window we can see the natural soil surface of the field outside.
Because life underground is generally pretty slow, it's difficult to see changes occur. Malcolm and his research students record their observations using a special camera that takes one picture every 4 seconds. This allows them to see a speeded up version of what's happening right under their feet. It was observation, this time of environmental damage visible in his own garden, that led our first finalist, Stephen Dyke, to begin his investigation into the effects of acid rain on seedlings.
Stephen Dyke was particularly well motivated. He was an individual very interested in ecology for quite some time before this project. We were impressed by the way in which he'd both written the report and set up the research in the first place.
Having a good background knowledge of the subject you're investigating can be a great help. Research, using reference books, newspapers, magazines and scientific reports, helps you learn from the work other scientists have done. Stephen found that this helped him to plan his investigation and experiments more thoroughly.
The books and journals are requested, please. There's a local British library nearby, and they're very good in supplying me with all the information I need. So I just fill in forms about acid rain, and they'll just bring journals, books, previous studies all about the topic, which I can read on before I go into the experiments myself.
When we knew that Stephen was on the short list of finalists, we sent Nick Davis to tell him the good news.
Stephen, I've got some good news. You are one of the finalists in this year's Young Ecologist of the Year competition.
Excellent.
Well done, congratulations.
Thanks.
Is this the experiment here?
This was the first experiment I did, a very basic experiment just to get the general feel of what acid rain can do to plants. It's simply lettuce seedlings. This was the one which I gave water to, and this is the one which I gave the synthesized acid rain to.
Golly,
as you can see, it's really been destroyed.
The effects of acid rain were first noticed in Scandinavia 30 years ago. In the lakes, the fish and plants are dying, and so too are the forests. They've blamed this on the sulphurous smoke belching from Europe's power stations. Whilst Britain is trying to cut down on these emissions, scientists question whether it's really enough to stop the damage. As you can see from this crumbling stonework, acid rain destroys not just plants, but buildings as well. Stephen collected samples of rainwater from Leeds city centre and used these to help him create his own synthesized acid rain. Stephen's first experiment supported his hypothesis that acid rain kills plants, and he wanted to investigate further to see how this might happen. His new hypothesis was that as rainwater evaporated and transpired from leaves, the concentration of the acid would increase and cause physical damage.
This was the one I'd added with water. As you can see, they're growing rather nicely.
Mm-hmm.
That one here was the one I'd sprayed with the acid rain.
Gosh.
And as you can see, they're in real trouble.
Little brown marks on the leaves,
yeah,
which are absent from the other ones.
It's important to control experimental conditions very carefully to eliminate any variables which may stop you from getting reliable results, and working in a lab can help. But it isn't always possible to recreate a set of conditions inside a laboratory. That's where the Rhizotron has been useful.
A lot of people have been interested in the factors that affect the growth of plants in the field. And many people have looked at things like light and temperature and fertilizers. And these are all things that you can manipulate in the laboratory to see the effects. But we are interested in what the effects of the small animals that live in the soil are on plants. And that, of course, is a field problem and is much more difficult to study.
Gardeners have always valued earthworms. Now scientists are also becoming excited by the help worms can provide in the fight against soil pollution. 3 years ago, a fire at a plastic recycling centre caused tremendous environmental damage. It sent a lava stream of molten plastic flowing into the neighbouring woodlands, where it then solidified. It's still there today, though soil is now starting to form on its surface. Biologists are using worms to check how much pollution from the plastic is seeping into the landscape by taking harmless blood samples from worms around the site. Because worms can save scientists time and money, their detective role is going to be much used all over the country. Our next finalist also had a special interest in worms. Ian Rushton discovered that it was difficult to study worms in their natural habitat, especially if the ground they're in is very hard. We asked Michaela Strachan to take a break from the Really Wild show
Spotted him.
and go back to nature to let Ian know what the judges had decided.
Hello, Ian. Hi. I've got some good news for you. Yeah? You're a finalist in the competition. You're off to the conference. Congratulations. Thanks. So this is a bit hard, is it?
Yeah.
Shall we go?
Yeah.
Okay. Gosh, it is, isn't it? So you gave up on this idea.
Yeah.
What did you decide to do next?
Well, I decided instead of this to simulate it in the lab using empty ice cream tubs.
So you just had to modify your original idea, which
Yeah.
is very important when you're being a scientist, isn't it?
Yeah.
Ian's hypothesis was that soil with worms in it would be better aerated than soil without worms. For his investigation, Ian used 6 plastic tubs to simulate 3 habitats in which worms are found: plain soil, leaf-covered soil, and grass-covered soil. For each of these different environments, Ian had a control tub containing no worms.
Right, I'm going to do the water percolation test now. I'll screw the bean can in as far as it goes. This is the one without worms, and can you stop the clock when I say?
When you say so.
100 cc's of water, go.
The first test was to see if water drained more slowly through the soil in which there were no worms. The second experiment tested how far an object could penetrate the soil using a penetrometer. This ensures the object falls the same distance every time and controls where it lands. Ian then recorded the results of each test before repeating the process. If his hypothesis was correct and the test was conducted fairly, then the object should penetrate further into the soil that has worms in it. And the results of the penetrometer test?
Well, I found out that it didn't prove my theory because I don't know what the reasons were. Maybe the tests were done at different times because I watered the tubs
right
to keep them wet and moist.
Not all tests turn out how you hope
No,
they do, they?
no.
What about the water test?
Well, that turned out how I predicted.
Which was?
That the soil will be more broken up.
With the worms in it?
Yeah.
So Ian, you obviously know a lot about earthworms now.
Yeah.
How do they help the soil?
Well, they break it down a bit and they break down the particles so the plants can take them up through their roots easily.
So it fertilises the soil,
Yeah.
really, doesn't it?
Yeah.
And have you been pleased with your experiments?
Yeah, I've been fairly pleased, yeah.
How would you improve them if you did them again?
I'd probably make it more of a fair test by making sure I did the penetrometer test at the same distance of time after I watered them.
He was a finalist because he had used good scientific methodology in his approach. He'd used controls.
For experiments to have any meaning, to be able to draw conclusions from the tests, scientists have to record the results. Here at the university in Bangor, just as in many research institutes all over the world, they collect their data and analyse it on computers. Now Malcolm, what happens to data from the Rhizotron?
Well, the form of the data, much of it is coming in from the cine camera, because using a time-lapse camera allows us to watch things that take a very long time in a short time. So we use the computers to help us analyze the pictures frame by frame, to count what animals are there, what they're doing, who is touching whom. And once we've got all this material in the memory of the computer, then of course there's lots of things we can do with it. We can actually use the computer then for looking for patterns in the data. Are there patterns over time? do different animals, different species of animals, take a different length of time to locate their prey. And also, do different species of animals hunt at different depths. So it may be that your safety depends upon how deep you are. In all these things, computers are immense help to us.
As electronic equipment becomes cheaper and more robust, so it's possible for many more scientists to have access to the same kinds of equipment that university researchers use. One major piece of equipment is a data logger. This enables computers to record actual readings from sensors. 5 girls from Devonport High School made good use of a data logger in their investigation. And as well as recording this numerical data, they used a video camera to help them record the conditions around their test and control sites. To tell them the good news that they were finalists, we sent Blue Peter's Claire Bradley.
Well, I think this is the right room, and there's 5 girls in there that I want to talk to.
Branch, and then give it a big bang with a stick.
Mr. Edwards, I'm looking for Sharon, Melanie, Katie, Anna and Laura.
Oh, there they are, there, that's 5.
Hi! Well, congratulations. You are finalists in the Young Ecologist of the Year competition. So tell me, what was your project all about?
Well, it was an investigation to find out the order of recolonisation by plants and animals in 2 different areas. One of the areas was bare soil and one was burnt.
Huge areas of the world's forests are destroyed by fire every year, both naturally and more worryingly by people who want to clear the land for farming. Such large-scale destruction deprives the earth of its lungs, the trees. They release oxygen into the atmosphere and absorb carbon dioxide, and this burning adds to the problem of global warming. The effect on plant and animal life is disastrous. Millions of species lost forever. Rainforests turned to farming land will never be the same again, but where the cause of the fire is accidental, how long will it take before the land returns to the way it was, before it can recolonise? You don't need large areas to test the rate of recolonisation. Let's see how the girls from Devonport went about their investigations.
This is a nice wildlife area. Yeah. Are you using the Stevenson screen as part of your experiments?
Yep, we're using it to hold the data logger, which collects all the information. Over the Easter holidays, we set up a data logger to record the temperature of our 2 plots, the air temperature, and then the light. And we told the computer to record them each day, and we did that at 3-hour intervals.
Well, here are the plots over here, I can see. Now look, this one is obviously the burnt one.
Yes, but you can't see all of the area it's burnt because the vegetation seems to be growing from the outside edge inwards and comes all the way back to about here, where it was burnt originally.
To help them in their investigations, the girls used several different experiments.
We did agar plates to find out the bacteria in the soil, and we did several soil tests to find out the potash and the pH level of the soil.
They also conducted a test to discover what animal life there was in the soil. The heat and bright light from a lamp placed over the soil sample causes any animals that are there to burrow deeper. Eventually they'll fall out of the bottom of the funnel and land in a beaker of alcohol where they can then be recorded. This apparatus is called a Tullgren funnel. So what conclusions did they draw from the test results?
We thought that in the Tullgren funnel we find more animals, but we only found 2.
When we burnt the actual area, the potash and the pH level were changed, so the animals didn't like the area, and so they didn't recolonise there.
The pH level of the unburnt was 6, and the pH level of the burnt area was 8, so it was more alkaline. Some of the plants which are grown on the unburnt area won't grow on this area, and we think it might be because they only grow in a pH of 6.
Maybe. This is why your experiment is so interesting, you see. Well, let's go and have a look at the other ones. There's a lot more plants there. Now, how did you go about clearing this area of vegetation?
Well, last year initially we dug it over using spades and forks, but it grew back very quickly and we didn't expect that. So this year we dug it over again, and this time we trampled down the ground, so it was a lot firmer.
So you didn't quite realise how long seeds can actually live in the soil, because seeds can live for up to 80 years. And not all the plants obviously live quite as long as that. That's quite an exceptional case. But up to, 20 or 30 years. So what do you think is the most important thing that this project has taught you?
We expected different plants to go back on the unburnt and the burnt area, but I don't think we expected such a difference, because there's hardly any plants at all in the middle of the burnt area, where there are lots of variety of plants on the unburnt.
Well done. I think it's a very interesting project, and I wish you lots of luck with the final.
Thank you.
They thought of a particular problem. They set about finding a way to solve it. They set up an experiment which would give them a definite answer, and they carried it out meticulously using various techniques, which would all form part of their work for the national curriculum.
Well, the tension's visibly rising here as the time for the announcement draws near. But first, I've got to go and get changed. So who will win the competition? If we look at the way each of the finalists carried out their investigations, their methodologies, we can see that they have certain things in common. We've seen how important it is to look at what's happening around you, to see what might be worth investigating, and then to look at what you've chosen very closely. But it can be a great help to find out more about a subject, so you can plan what exactly you'll do and decide how you'll do it. How sometimes you'll have an idea or hypothesis you think explains your observations, but at other times your hypothesis comes later as you start your tests, the heart of every investigation. Making sure that safe and fair testing is carried out to give reliable results, you can then collect data. Of course, for tests to have any meaning, you need to record the data clearly and accurately, so that when you've finished, you can do what you set out to do at the start, to draw conclusions, pulling together all the evidence you've collected and telling everybody about it, just like our finalists have done.
It gives me great pleasure to be able to announce tonight the winners of the Young Ecologist of the Year competition.
The judges awarded the runners-up prize to Ian Rushton from Lancaster Royal Grammar School. And because of the high standard of their entries, they awarded 2 first prizes, to Katie, Melanie, Anna, Sharon and Laura from Devonport High School for Girls, and to Stephen Dyke from Boston Spa Comprehensive. Some excellent work and a fitting conclusion to 3 truly scientific investigations.